Thank you. So thanks for attending the talk. It's great to see such incredible support for a truly multidisciplinary problem. And so when the organizers first asked me to give this talk, my immediate first thought was, what's a better treatment for insomnia? A talk on molecular mechanisms of cardiotoxicity or 5 of Ambien? And so hopefully at the end of the talk you'll find it to be informative and more importantly you'll be awake. So when I think of cardiotoxicity, and cancer, I usually think of it as a model of competing risks. And so here's data from the Physicians' Health Study. And so this took 22,000 male physicians and followed them over several decades and basically looked for the incidence of cancer and cardiovascular disease. And so what you can see here is the blue line is the incidence of cancer. The red line is the incidence of cardiovascular disease, and you can see that both of these are diseases of aging. And so it doesn't take a lot of rocket science to understand that really these are diseases of competing risk. So if you survive your cancer, you're at risk for cardiovascular disease. If you survive your cardiovascular disease, you're at risk for cancer. But more importantly, I think that's something that's emerged is that actually cancer therapeutics can interact with your risk for cardiovascular disease, and that's really what we're going to focus on today. So most of the time I spend thinking about cells and biology, and when I think of the heart, I think of it as a composition, an organ that's made up of a complex number of cell types. And so there are a variety of cells that have important functions. And I think to try to understand how various therapies interact with the heart, one needs to actually think about the cells that are involved, and that'll help to understand how they have different effects. And so the heart is made up of cardiomyocytes, which are the cells that are the major contractile function of the heart. There's the outer epicardium, which is a mesothelial sheath that covers the heart. And this is a cell source that's important for providing important growth factors to making sure the heart is healthy. You've got the blood vessels that line the heart, and we know those are important. And the endothelial cells are the cells that actually make up the blood vessels. And then finally, you've got these fibroblasts, and the fibroblasts are scar-forming cells. And so more broad therapies actually tend to have broad effects on all of these cell types. For example, you just heard about the effects of radiation therapy. And radiation toxicity of the heart has very broad effects, in large part because it affects everything. So if you've got cardiomyocytes, you can get a stiff ventricle. If you affect the epicardium, you can get a pericarditis or a constrictive pericarditis. You can trigger scar-forming populations. And then if you affect the endothelium, you can get microvascular disease and even overt epicardial coronary disease. And I think as therapies have moved forward and we've got more and more targeted therapies for various malignancies, you're starting to see differential effects on these cell types. And as a result, you get these very differing profiles of cardiotoxicity. And so the way I will break this down is to actually go through a couple of the cell types and some specific examples. And so when people think of cardiotoxicity, the classic model is to think of a type 1 and type 2, and with type 1 being this irreversible cardiac damage. And so I think the way to think about it is you get a cardiomyocyte and then you injure it in such a way that you actually have death of the cardiomyocytes. And so what you have left over is a big hole, and you scar, and the ventricle deteriorates. And so classically we think of type 1 toxicity as this irreversible cardiomyocyte damage. And really the prototypical agent to affect type 1 cardiotoxicity are anthracyclines. People have really worked a long time. This is no mystery. Anthracycline toxicity has been around for decades, and there's been a lot of intense research to try to figure out how exactly it works. And basically the answer is it's very complicated. It's very difficult to know. And what I mean by that is this is the state of the art, this figure. It's very challenging. And so as you know, anthracyclines are associated with an acute phase cardiotoxicity that can result in a myopericarditis, but it's really this chronic phase that we tend to see later out, and that can be quite insidious and quite deleterious. And the way people now think of to summarize how people think of this, the mechanism of action, is that anthracyclines like doxorubicin actually create a stress, an oxidative stress on the heart. And so basically, if you can make it out, you get this oxidative stress and you get these free radicals that are made, and then you damage cardiomyocyte membranes. You damage important receptors that are critical to making the heart work, like the beta-adrenergic receptor. You can poison the mitochondria so that you uncouple the electron transport chain from oxidative phosphorylation. And that's pretty bad for the heart because the heart's really a bag of cardiomyocytes that contain mitochondria. And so you're basically energy starving the heart. And then finally, all the same mechanisms that can kill cancer cells can also affect the heart as well. And so in malignancies, one way that anthracyclines are thought to work is by inhibiting this topoisomerase, which is responsible for creating, keeping the fidelity of DNA. So if you impair topoisomerase, you get more DNA damage and can trigger apoptotic responses. In a cancer cell, that's good. You kill cancer cells. In the heart, that's not so good. You can't really make new myocytes very, very well. So if you kill cardiomyocytes, that's a problem. And so, again, it's quite complicated, but the take-home message here for type 1 toxicity is that you get irreversible cell death and that you have reactive oxygen stress. The second type of cardiotoxicity is another way to make the heart weak. So rather than kill the myocytes, what you could actually do is just make them not work very well. And so for that, the prototypical agent is actually trastuzumab. And so again, you're taking the myocyte, they're actually alive, but they're just not working so hot. And so the mechanism for that is a little bit more straightforward. And so trastuzumab is commonly used to treat breast cancers. And the way it works is it works by targeting the neuregulin ERBB axis. And so neuregulin is is actually a potent mitogen for many cell types, so it makes things grow. For cancer, again, you want to block the growth pathway, but it has very strong effects on the heart. So if you knock out in animal models, you have defects in any one of the receptors for neuregulin or its receptors, you get this very strong developmental defects where the heart just simply does not develop. You get really a paucity of cardiomyocytes. Postnatally, if you impair ERBB axis, the heart's not can't adapt to stress. So if you stress the heart, it's just going to crump out on you. And so it's not surprising that therapies that target the ERBB2 receptor, in this case trastuzumab, actually can potentiate the heart. And so if you stress the heart in these situations, you can lead to cardiomyocyte dysfunction. And this is, again, a complicated pathway involving MAP kinases, et cetera, et cetera. What's interesting about this particular mechanism of trastuzumab is that other small molecule inhibitors of ERBB don't seem to have the same cardiotoxicity, and exactly why that is not known. So before I move further, we should just take a step back and on this concept of type 1 and type 2 cardiotoxicity and realize that in the real world it's not so simple. There's a lot of overlap, and that may be a bit of a simplification. So classically, type 1 cardiotoxicity you can't get better from, but in reality we know now from large data sets that if you actually identify early LV dysfunction in patients treated with anthracyclines, you can actually recover the ventricle. Similarly, if you let trastuzumab toxicity go too far, you can actually still see scar form in the heart, which might be a sign of irreversibility. Okay, so let's move on to some of the the newer agents. And so in the last 20 to 30 years, there's been a boom in small molecule and medicinal chemistry. And this has allowed for development of small molecules that can target tyrosine kinases with varying degrees of specificity. So I'm not a medicinal chemist, and so I'm going to give you my interpretation. So these are a lot of different tyrosine kinases. They're treated for lots of different things, and I honestly can't really tell the difference between any of these things. They kind of look alike, and I make that point because the 3D conformation of these are very similar. And while our intention is to really focally target specific targets, it makes sense that they're going to be off-target effects. And so if you broadly inhibit tyrosine kinases, you're probably going to get off-target effects. And I think that's where a lot of the toxicities occur in this class of drugs. So if you take, for example, the tyrosine kinase inhibitor sunitinib, it's a very broad tyrosine kinase inhibitor with very potent anti-cancer effects, but it can actually be quite potent and deleterious to the heart. And so if you follow patients with sunitinib, about 10% of patients will actually develop clinical heart failure. A quarter of patients will have some decrement in LV dysfunction. And you can see from this diagram, taken from this review, that the way this works is it actually inhibits a lot of important tyrosine kinases that are quite critical to homeostasis in the heart. And so taking one step away and moving away from the myocyte, if you focus on another cell type that's really important to the heart's homeostasis, the endothelium, that's actually that's another important target for cancer therapies. And certainly cancers need blood to grow, and so it's very intuitive to target the blood supply, but similarly the heart needs blood to work as well, and so it's not, again, rocket science to think that there may be some off-target effects. So again, the endothelium are the cells that line blood vessels. Their purpose is to keep blood vessels open, so it's a balance of making clots and not making clots. And their also purpose is to keep the blood vessels open by regulating vasomotor tone. And so various agents can actually differentially affect the endothelium and predispose to thrombosis and to hypertension. So for example, bevacizumab, cyclophosphamide, and thalidomide are drugs that are associated with thrombosis. I'll never forget as an intern seeing a woman who was treated with thalidomide coming with acute bilateral lower extremity emboli, had bilateral cold feet, so it can be quite fulminant. And then similarly, by interfering endothelial homeostasis and decreasing nitric oxide, you can get hypertension. And in fact, clinically, this is what we see a lot. We're starting to see a lot of patients treated with tyrosine kinase inhibitors have actually this very, very potent and malignant hypertension. And so I just put this here for your reference in case you're interested. But again, the major cardiotoxicities associated with TKAs are ventricular dysfunction, thrombosis, hemorrhage, and hypertension. Okay. So is this a hopeless field? Is there something to do? And I actually think this is great. Now, as tyrosine kinase inhibitors are getting better, as small molecule development is getting better, one of the in addition to just the cancer effects, I think a important outcome measure is going to be minimizing these off-target or cardiotoxicity, since it has such a profound effect on long-term outcomes. And so as we look to the future to try to minimize cardiotoxicity, I think it starts at a preclinical level, which better screening, trying to find molecules that have less off-target effects. And two is trying to understand how these molecules are actually causing cardiotoxicity and trying to come up with specific antidotes. And so I'm going to just spend the remainder of the talk talking about two ideas to really focus on this. So back in 2012, Shinya Yamanaka came up with this idea of induced pluripotent stem cells. And he won the Nobel Prize for it in 2012. And basically what one can do is you can take a skin cell, you can put a cocktail of genetic factors, and you can make an embryonic stem cell. And so this was thought to be pie in the sky, but it turns out that you can take an individual person's cell and then get heart muscle cells. You can get a variety of different cell types by differentiating these things. And so in the past 10 years since this was discovered, people have used these in very creative ways to try to affect a personalized type of paradigm. And so people study these as models of developmental biology. They use them as to study replacement therapy. But recently it's actually become a platform for drug discovery. So there are many small companies that are actually taking patient panels of iPS cells, making them into cardiomyocytes, and selling them to pharmaceutical companies and saying, you should use this as a way to screen for cardiotoxicity. And so this is an emerging field, but hopefully a mechanism by which we can actually screen compounds in the early phases to see if they're actually going to have deleterious effects on the heart. The second way I think that there's a future to sort of preserve anti-neoplastic effects but minimize off-target effects is through targeted antidotes. And so because this problem has emerged to be such an important issue in treating patients with cancer, it's actually become an important issue as well for preclinical research, and is starting a new field of therapies trying to minimize cardiotoxicity. And so here's an example of one such study done by Randall Peterson at Mass General. And so what they did was they took zebrafish and they made these poor embryos swim in a bunch of doxorubicin, and they found that the hearts don't work very well. If you stain for a marker of apoptosis by tunnel staining, that's these green cells, you see a lot of cell death in these hearts. And so they did a drug screen, and they took a bunch of off-the-shelf chemicals and tried to see what's going to rescue this phenotype. Can we minimize the apoptosis in response to doxorubicin? And so they came up with two that they found, visnagen and diphenylurea. These molecules look nothing alike. They didn't know if these things were anything related to each other, but it turns out if you study them a little bit harder, they both actually preserve mitochondrial function, so sort of makes sense with what we talked about before with doxorubicin. And then as a proof of concept to see if it actually works in mammals, they went to a mouse model of doxorubicin-induced cardiomyopathy. And so again, you can see that animals that are treated with doxorubicin tend to have a decrease in fractional shortening compared to control animals. Their strain rates are decreased as well. But if you give them this drug, visnagen, that you can actually partially rescue the decrement in fractional shortening and strain rate. And so this is a new area. It's just really just the tip of the iceberg. But one could imagine that in the future there really will be a set of targeted antidotes that go along with specific chemotherapeutic agents for patients that experience cardiotoxicity. So with that, thank you for your attention. It looks like most people are awake, so really thank you.